Hello, and thank you for tuning in. My name is Lena Trodeshow. I am a research scientist at the Duke University Center for WashAid. At the Duke Center for WashAid, we focus on decentralized, non-sewered sanitation systems to address some of the sustainability and safety concerns for the billions of people around the world who lack access to safely managed sanitation. And we often find ourselves doing a delicate balancing act between safe sanitation and safely managed sanitation and issues regarding water scarcity. In addition to all of that, or as I've shown it here, below all of that, we have a responsibility to make sure that the technologies that we develop are appropriate for the cultural context and meet the needs and desires of our users. Because technology at the end of the day is only as effective as it is willing to be adopted by the people who it is intended to serve. Our approach to these problems focuses on user-centered design through post-flush technologies. And there are several reasons for this. One being that we want the users to be able to maintain their current behaviors. We also want to be able to connect to existing plumbing fixtures like squat toilets or pedestal toilets. And we also want to meet user preferences and aspirations for flush toilets. And this has been shown to be the case even in regions that are particularly water scarce. All of our technologies involve some aspect of solid-liquid separation as well as solids treatment. I'm not going to talk about these today. Instead, I'm going to talk about liquids treatment process, which makes up over 99% both by volume and weight of the toilet effluent. And the main concerns here, in addition to pathogen elimination, are also the environmental requirements, such as pH, nutrient content of the effluent, that we need to meet in order to enable water reuse or safe discharge to the environment. We currently have a household-scale liquid treatment prototype that aims to achieve water reuse through non-biological treatment processes. The reason for that is that biological processes typically have large footprints. They can be very susceptible to shock at small scales, and some of them even require inoculation or long startup times before they can reach their full treatment abilities. So our prototype consists of three main processes. We have ultrafiltration first, which removes suspended solids, followed by a granular activated carbon or GAC filter that removes soluble organics, and finally an electrochemical disinfection step, which also provides a residual chlorine for the water that is stored in the storage tank prior to reuse. And over here we have a snapshot of the influent black water coming into our system as well as the treated effluent. And what the chemical composition looks like for nutrients in this is we have about 100 to 250 milligrams per liter of total nitrogen and 50 to 80 milligrams per liter of total phosphorus in the black water influent. In the effluent, we do see a reduction 40 to 60% in the total nitrogen, as well as 30 to 40% of total phosphorus. And this is primarily due to the ultrafiltration removing the organic components of total nitrogen and total phosphorus. However, the inorganic nutrient components, primarily ammonium and phosphate ions, are still too high in the treated effluent for us to reuse this water for non-potable purposes or to discharge it safely into the environment. To improve the nutrient removal capabilities of our prototype and enable water reuse, we've been looking at silicate-based minerals that specifically target phosphate and ammonia. And the materials I'm shown here on the right, we have polonite, which is for phosphate removal, and then clinoptilolite for ammonium removal. And these materials are relatively inexpensive. They're non-toxic aluminosilicate minerals, and they have the potential to be regenerated and reused multiple times. Where that regeneration ability comes from is the different sorption mechanisms that are occurring on the surfaces. So these are dominated at the pH that we're interested in, around 7 to 8 for black water, primarily by electrostatic attraction as well as ion exchange mechanisms. And both of these, while being pH dependent, are reversible. So we can swing the pH one way or the other and have ions desorb from the surface, depending on the material we're talking about. And that can not only enable reuse of the media itself, but also gives us the option to potentially recover the phosphate and ammonium nutrients, which can be used as fertilizers. So the research questions I'm going to address today are listed here. First, where in our liquid treatment process should we utilize these sorbent materials to get the most out of them? Second, what are the capacities of these materials for use with black water? While they have been tested for other types of wastewater, black water is a really complicated mixture with a lot of potentially competing ions that could affect the sorption capacities. Third, can we regenerate these materials after use with black water so that they can be used multiple times? And finally, can we move these media from bench scale experiments to something more representative of how they would be used in a pilot system, so in a single pass configuration and at a larger scale? Most of what I'm going to show you today are the results of batch experiments, and these were performed using an orbital shaker. To do these, we have our flask, we have our media in there, and then we add in our black water, either raw or with some level of pre-treatment from our prototype. We then allow this to mix, and then we sample the liquid fraction and calculate the amount of ions, shown as the red dots here, that were sorbed onto the material. We can also do desorption experiments where we have our media that's loaded with ions after exposure to black water, and then we add a regeneration solution. In the case of polinite, this is a strong base, potassium hydroxide. In the case of clinoptilolite, this is HCl. We then sample the liquid fraction and measure the amount of ions that come off the material into the solution, so the amount that's desorbed. And to measure the concentrations of ammonium and orthophosphate, we're using colorimetry or UV vis spectrometry using a hot colorimeter. The first question we wanted to answer was where in the liquid treatment process we should place these minerals. I'm showing here data comparing loading of the materials to the percent removal for either phosphate in the case of polinite or ammonia in the case of clinoptilolite. And what we see immediately is that for polinite, we get the worst performance of this material when it is exposed to raw black water, but the performance improves quite a lot after the black water has been through ultrafiltration and the GAC column. And there's a slight dip in performance for the black water after it's gone through the entire process, including the electrochemical disinfection step. What was interesting and kind of surprising is that the clinoptilolite performed the same regardless of how many steps in the treatment process the black water had been through, and actually performed quite well for the raw black water as well. So the takeaway from this is that we decided that we were going to install these after the GAC column, but before the EC cell. We then performed several different experiments to look at the overall sorption capacities of these materials for their target ions in black water that was pulled after the ultrafiltration and GAC column. And what we found was that for clinoptilolite, we measured on average about 12 milligrams of ammonia per gram of media capacity. And for polinite, we measured a capacity of about 8 milligrams of phosphate per gram of media. And since both of these materials were off the shelf from commercial suppliers, we compared that to what the listed capacity was that we received from the manufacturers. And what we saw is that for clinoptilolite, the range that we measured of 12 milligrams per gram was right within the range of what we would have expected. But what was surprising is that for polinite, we only measured about an 8 milligram per gram capacity, whereas the manufacturer had listed a maximum capacity of 120 milligrams per gram. And this really drives home a point that we focus on a lot in our research, is that testing different components with real wastewater is critical for design of full-scale systems. And that's why we, even though these are off-the-shelf components that have been tested with other wastewaters previously, it's really critical for us to put them into our prototype at the place that they would be in our liquid treatment process to see how they behave when exposed to black water. Next, we're going to look at results for our experiments to try to regenerate and reuse these materials over multiple black water exposure cycles. And these are kind of mixed results. We found that in the case of clinoptilolite, we were able to get 70 to 100% of the ammonia to desorb when we exposed it to a 1 molar solution of HCl. The desorption was a little bit lower at 0.1 molar, but by increasing that concentration of the regeneration solution, we got much better desorption. That was not true in the case of phosphate. Regardless of the strength of the regeneration solution, we were only able to get about 20 to 30% of phosphate desorbed from the polinite. And there's a couple of different things that indicates. The first one is that these electrostatic and ion exchange interactions are probably not the dominant mechanism. They probably only account for about 20 to 30% of the phosphorus sorption. So that means that phosphate precipitation is probably occurring inside this material. That means that there's going to be a finite capacity. As we continue doing more regeneration cycles here, we would start to see this percent removal dip and eventually plummet, and we wouldn't be able to reuse this material multiple times. So that has important consequences when thinking about in what kind of context we would want to use these. So maybe clinoptilolite with a good regeneration process could be used in low and middle income countries. But in the case of polinite, this is going to basically be a consumable, and we're going to have to think about in what context it makes sense to use it in that way. Moving on from batch experiments, we wanted to test whether or not these minerals would be effective integrated with our prototype. So I have a block diagram here on the left, but I'm going to be pointing at the photo here on the right to show you where the components are. So we have our storage tank that feeds by gravity into what we're calling our feed tank, and this is where we take our influent samples. That's connected to the ultrafiltration membrane, followed by the GAC column. And then instead of allowing the liquid to directly flow into the electrochemical tank as originally designed, we've hooked up these two tanks here. One of them is filled with 8.5 kilograms of clinoptilolite, and the second one is filled with just over 7 kilograms of polinite. And then that outlet from the polinite tank then feeds into the electrochemical cell. And what you can't see here on the floor is there's a treated water tank, and that's where we pump the effluent out to, and that's where we sample our effluent from. The flow rate through the system is approximately 100 to 150 milliliters per minute in this configuration. And I just want to mention that the size of these tanks was not chosen because they are optimal for any particular set of circumstances. We had these in the lab, and we also had identical ones at our field testing site, and so we wanted to make it easy for them to install this, so we decided to use the tanks that our field partners had available. So when we turn on this system, we see a couple of interesting things. Here in gray, you can see data points where the nutrient removal tanks were not connected to the prototype. So that's sort of the baseline removal of nitrogen and phosphorus. And then in the colored data points, you can see when the tanks are connected, how the nutrient removal improves immediately upon connecting these tanks. And then you can see again, as we disconnect the tanks, the performance suffers, and then it recovers immediately once the tanks are reconnected. Adding a little bit more media gives us a little boost because we have more sorption sites available. But what's really nice about this is that this is our first experiment trying this at scale. The sizing of the nutrient tanks is completely not optimized, but we're already getting very close to some of the percent removal standards that are set, for example, by the ISO 3500, which dictates water reuse for non-sewered sanitation systems. They require 70% removal of total nitrogen and 80% removal of total phosphorus, as shown by these dashed lines. And so in this unoptimized experiment, we're very close to hitting that, and the performance is fairly level across the range of the experiments that we did here. One problem is that the effluent pH is quite high. It's above 9 for all of these experiments where we're connected to the nutrient tank. And that's due to the basic nature of the polinite and releasing hydroxide ions into the solution. A final point of interest is that we observed no loss in performance of either of these materials after over 12 weeks of inactivity. Our lab was shut down for mid-March through late June due to the COVID-19 pandemic, and upon coming back and starting operations, we ran a few cycles with the prototype without the nutrient tanks connected, and then as soon as we connected those tanks again, we saw the performance kick back on and great removal of both ammonium and phosphate. And this is interesting. We're thinking about the potential to use these materials in contexts where the toilet use itself is intermittent and not constantly receiving a supply of fresh material. To summarize, we found an appropriate place in our liquid treatment process after the ultrafiltration unit to utilize these materials. We also were able to measure their sorptive capacities for use in black water. We did do some experiments looking at the regeneration potential. It looks like clinoptilolite is a promising candidate for regeneration, while polinite maybe not so much. And finally, these are effective in a single pass type of configuration, and they are also not affected by intermittent use. However, the high effluent pH may be limiting for certain use cases. So we're currently looking at field testing clinoptilolite. We decided not to move forward with polonite because of the high effluent pH. However, this work has been delayed due to the pandemic. We're also looking at how to recycle and determine if there's any value in the regeneration solutions, perhaps as a fertilizer. We are looking at new materials, new synthetic materials for phosphate removal. And then we're looking at how to scale up clinoptilolite for something like a toilet block on the scale of 1,000 liters per day processing. I'd like to thank Mariana and Kayana, who are fantastic students who did a lot of the work that you saw here today. I'd like to thank the Bill and Melinda Gates Foundation for funding through the Center for WashAid, and I'd like to thank you for listening.